that the recording will work, and then what we'll do is we'll transcribe it, and then what we'll do is we'll send it out to everyone via email, and we'll also post it to our website. So without further ado, I'd like to send it over to Dr. Jett and Dr. Spriggs.
Okay, so good afternoon, everyone. It's pretty exciting. We've got near 100 registered participants in the webinar. I'm Dr. David Jett. I'm director of the NIH CounterACT Program, and I'm joined by our program manager, Dr. Shardell Spriggs, today to present a webinar entitled Current NIH CounterACT Funding Opportunity Announcements, webinar for potential applicants. So what we will do is we will present the slides and then take questions, as Tim mentioned, questions that were submitted or any new questions at the end of the webinar. So please keep in mind that some very specific questions may be best done on a one-on-one basis with NIH staff at a later time. Okay, it's not done. All right, some technical difficulty. This one? Okay. All right, little technical pause there. So, webinar goals are to provide a description of the NIH CounterACT program, talk in some detail about our specific funding opportunities, and then try to provide some tips and lessons learned for for folks who are thinking about applying to the program. So the program has supported research since 2006, and its mission is to understand fundamental mechanisms of toxicity caused by these chemical threats and apply that knowledge to develop promising therapeutics. Overall, all goals are to improve the nation's medical response capabilities, support cutting-edge research to improve our knowledge base, and develop optimized lead compounds for potential transition to advanced development. So I guess the most common question we get is about the chemical threats we need studied. Presented here are broad examples of chemical threats of interest. They include traditional chemical warfare agents such as sarin and VX and Soman, chlorine, sulfur mustard, toxic industrial chemicals such as cyanide, hydrogen sulfide, and others, toxic agricultural chemicals such as insecticides like aldicarb, chlorpyrifos, parathion, and rodenticides like rodificom and tetramine. And then finally, we are interested in pharmaceutical-based agents, for instance, the opioids, fentanyls. And the program is guided by a list of about 200 chemicals developed by the federal government. And applicants should contact myself or Dr. Spriggs to confirm that the chemical or chemicals they wish to study are on the list before developing an application. We cannot at this time share that list with you, but we can discuss the list in detail with you to facilitate your choice or confirm your choice that it's on the list. One thing that is helpful and is not included in our funding announcements is our list of toxidromes. This is where we have grouped chemicals on the list based on their mode of action. This not only gives you an idea of the kinds of chemical threats, but medical countermeasures developed for one chemical may also be effective for another chemical threat in that category. And these toxidromes include things like anticoagulants, rodificom, blood agents like hydrogen cyanide and sulfide, cholinergic warfare agents that we discussed, cholinergic pesticides like parathion, chlorpyrifos, and phorate, and aldicarb, convulsants like picotoxin, TETs, and strychnine, hemolytic and metabolic agents like arsenic trioxide, gallium sulfate, and arsine, opioids, the fentanyls, diacylmorphine, lower pulmonary agents like chlorine and phosgene, and upper pulmonary agents like ammonia and sulfur dioxide. And then finally, the vesicants like sulfur mustard and phosgene oxime. So the program is truly a trans-NIH effort. Because of the diversity of chemicals and their effects, several other NIH institutes and centers participate in the program based on expertise needed to cover ocular, skin, pulmonary, and other effects of the chemicals we study. These include the National Institutes of Neurological Disorders and Stroke, Arthritis and Musculoskeletal and Skin Diseases, Environmental Health Sciences, Drug Abuse, the Eye Institute, and the National Institute of Allergy and Infectious Diseases that provides oversight and manages the contracts. Each IC has a program officer assigned that can help you with specific scientific questions. And we also rely on other ICs from time to time for advice, such as the National Institute of Child Health and Human Development, National Library of Medicine, and Heart, Lung, and Blood Institute. The overall NIH CounterACT program is designed to support research from the discovery phase, including the identification of therapeutic targets, through the development of optimized lead candidates ready for advanced development by other agencies like the Biomedical Advanced Research and Development Authority, or BARDA, or other entities. Our program includes several types of grant programs to be discussed later, and contract core facilities managed by the Chemical Countermeasure Research Program at NIAID. These contract programs support grant projects and can be used to generate preliminary data for grant applications. There are also NIH-supported interagency agreements with the Department of Defense laboratories that conduct research in this area.
The grant programs that we will discuss are progressive, starting at exploratory R21s through lead identification and optimization U01 projects that result in candidate therapeutics and major advances in our general scientific knowledge base in the literature. It is important to read the funding opportunity announcements listed here and talk to the staff at NIH to see specifically where you may fit among these programs. Please visit our website to see the latest funding announcements as well as a wealth of information including NIH scientific contacts, a list of funded investigators and publications, tips on writing milestones, core resources, and other information. Again, it is critical to use the latest funding announcement when composing your application since the entry requirements and review criteria may change from one version to the next. The CounterACT R21 funding opportunity announcement is intended for exploratory or developmental translational research projects. This often serves as an entry point into the program for many investigators, including new or junior investigators with promising project ideas that have little to no preliminary data. These are sometimes referred to as high-risk, high-reward projects. Currently funded investigators often use this mechanism for follow-on studies that are out of scope of their project, such as validation in vulnerable populations. The goal of the R21 is to generate preliminary preclinical, screening, and efficacy data to facilitate the development of competitive applications to more extensive support, such as the CounterACT U01 mechanism. The scope includes, but is not limited to, the creation and validation of screening assays, animal model development, identification of therapeutic targets and biomarkers, and the development of proof of principle efficacy data for candidate therapeutic compounds. Each of our funding opportunity announcements has one receipt date per year. Shown here is a schematic of the next funding cycle for the R21. The application receipt date is May 28th, with review in October, and awards will be released in March of next year. The application budget may not exceed $275,000 in direct costs for the 2-year project period. with no more than $200,000 requested in direct costs in any single project year. Please keep this in mind when writing your application budget. The R21 is not renewable. Because there is only one receipt date a year, you will need to time your studies to make sure that you have enough preliminary data well before your grant runs out to apply for the next level of funding in the program. This will ensure that you don't have a gap year in your funding. Using the R21 in the example shown here, if you receive your award in March of 2020, you will need to apply for U01 funding 1.5 years into the award. I thought it was important to mention this here because we have so many R21 investigators that also apply for U01 funding.
Okay, so the U01s. So this is the next sort of level of research that we support. The first U01 supports research on the identification of lead compounds. So by the end of the 3-year project in this case, you should have a compound or compounds that is biologically active and synthetically feasible, where specificity, affinity, potency, target selectivity, efficacy, and safety have been established both in vitro and in vivo models. The scope includes, but is not limited to, mechanistic research that leads to a lead compound in 3 years, confirmation of targets. It's not starting from scratch, but it is an earlier program than the next U01 we'll talk about. It includes screening for candidates, demonstration of relevant in vitro biological activity and target engagement, preliminary proof of concept efficacy data, as well as ADME-tox and pharmacokinetic pharmacodynamic data. Dates here are September 2019, then reviewed in March 2020, and the award is made in July of 2020. So it's often encouraged to submit a letter of intent for these applications so that we can sort of help our review team gather the best scientific expertise. Also, so the budget is may not exceed $300,000 in direct costs per year, and the project period may not exceed 3 years. So this is a 3-year grant, and it's no renewals. So the next U01 is for more advanced studies where a lead compound is already identified, and the goal here is that at the end of the 4 to 5 year project is an optimized lead compound ready for advanced development activities, possibly funded by BARDA or another agency, that lead to regulatory approval and licensure. In this case, it is recommended that you seek expertise in drug development and regulatory approval. The scope includes efficacy in human relevant animal models that's consistent with the intended use in humans, PK studies with validated analytical, an acceptable safety margin and a lack of drug interactions, and development of stable synthesis and formulation. More specifically, we have included criteria that, if met, should be eligible for consideration by BARDA or other advanced development entities, keeping in mind that completion of this work does not guarantee BARDA or any other entity will fund the advanced development of your candidate, but in theory, it should be ready for consideration for the next phase. So the project should culminate with one lead compound with well-understood ADME, efficacy data in appropriate animal model relevant to civilian concept of operations, including the route of administration, and extended post-exposure treatment if necessary, if applicable for your candidate, and initial pharm tox studies. You should also have stable, scalable synthesis worked out and a preliminary regulatory strategy, including a target product profile and a commercialization plan. There's a note here that there is a preference for candidate therapeutics that have an additional clinical indication, in addition to the indication for chemical threats. And then finally, you should have intellectual property as well as, importantly, freedom to operate during the project period. So for developing competitive applications, we've learned that addressing each one of these criteria, perhaps in table form, is extremely helpful to the grant reviewers. A fictional example is presented here where you can see each one is addressed, featuring the status, say it's complete or proposed, and where to find the proposed studies in the application. This not only reminds the reviewers what to look for, but also tells them where to readily find it in an often long, complex application. So the general rule is to make your application informative and easy to read. Okay, so for this UO1 program, the receipt dates are the same for the other one. The difference for this program is that the budget and project period is larger. The budget may not exceed $500,000 in direct cost per year, and the project period, instead of being 3, is a full 5 years. So finally, we also do support large multi-component research centers of excellence. The first 2 things to consider for these applications is, 1, whether you have strong preliminary data, and 2, if you have a team of researchers with projects that fit together synergistically around a central theme. The center structure includes 1 administrative core for center management and operations, 3 to 5 interrelated research and development projects, up to 3 scientific cores if needed and well justified, and 1 research education core. And the education core should be budgeted at no less than $50,000 per year and focus on the overall goal of increasing the number and capabilities of medical countermeasure researchers by building on the strengths of the center investigators. Each center has program oversight internally with a CounterACT Center Steering Committee and externally with an external advisory committee that meets every year. So the scope of these center projects encompasses basic research described in the R21 program through that described in the lead identification and optimization UO1 programs. However, specific outcomes must be met depending on the type of application. For new applications, at a minimum, you should identify a lead compound by the end of the project period as defined and described in the lead identification UO1 funding announcement. For renewal applications, you should have at least 1 optimized lead candidate as described in the lead optimization program. And then for second or more renewals, a clear track record of developing products that were transitioned to advanced development during previous project periods must be established. And I just made a note here that, a table for these could work well here as well. The receipt dates are the same for the UO1 programs, and the difference for this program is that the budget is much larger, but the project period is the same. The budget may not exceed $2.5 million in direct costs per year, and again, 5-year project period.
Some recent changes at NIH need special attention. Please read the specific notices listed here on enhancing and implementing rigor and transparency in NIH-funded research. Pay close attention to describing scientific premise, specifically the quality, including strengths and weaknesses of the evidence from your lab and the literature. Be sure to address sex as a biological variable. If you cannot use both sexes or you see no need to, have a good explanation why not. Too expensive is usually not an acceptable reason. Be sure to address biohazards, containment and disposal of, as well as authentication of key agents used in your research, including the chemical threat agent. Saying you purchased something from Sigma or other supplier is not sufficient. You will need to run an analysis to verify the identity and purity of key compounds. The emphasis here is on key agent and does not include minor reagents and buffers. Do not use the vertebrate animal section for experimental details to circumvent page limits. The reviewers are not required to consider this section for that purpose, and this may result in less enthusiastic review scores.
So, some important CounterACT specific issues to consider are first, how the therapeutic you are proposing will be used in humans. This is especially important for the more advanced studies in the optimization and centers programs. If reviewers see no real-world utility, the rest of the review usually does not go well. We also encourage research with vulnerable populations such as pediatrics and pregnant women, and we also encourage working with drugs that are already approved for other indications, since sometimes the regulatory pathway to approval is less difficult. And finally, therapeutics that can be used both as a medical countermeasure and a more traditional indication are a priority. For example, an anticonvulsant used for nerve agent-induced seizures as well as epilepsy would be desirable, since the market for such a therapeutic would be much larger than one that just treats nerve agent exposures. So your research strategy should include the structure of your compound when possible, and you should look at FDA guidances for help with a regulatory strategy, especially the FDA animal rule guidance that can be found on our website. Milestones, they are used for all our programs except the R21. They should be based on results or outcomes, not simply that a task or study has been completed. We strongly advise you to look at our website for instructions on how to write effective milestones for these programs. For intellectual property, the goal is to show that you have freedom to operate during your project period and beyond, and a commercialization plan is also required for some of the funding announcements, and you should seek help from experienced collaborators.
This concludes the presentation part of the webinar. We would like to thank you all for taking the time to listen and hope it has been helpful to potential grant applicants. Here is a list of NIH staff that participate in the program. Their contact information is available on the CounterACT website listed here. Please feel free to contact any of us for one-on-one consultation. We will now address some specific questions we have received.
Okay, just to follow up on that, when I will be sending out these slides by the end of this week. The recording to have it transcribed, that'll take a little bit longer, but you can definitely expect to have the slides sent to you later this week. And I will make sure to include Dr. Jett and Dr. Spriggs' email address on there, because we definitely advise you to reach out if you have any questions. So without further ado, let's get started with these questions. If you just bear with me 1 second. Okay, so the first questions. There's a question about how UO1 PIs and co-PIs are recruited or organized as a team?
Well, I guess as you know, we don't, the way we recruit people into the program is just by advertising the funding announcements. The teams, in terms of the teams, I would just first figure out what you'd like to do and what you would need to accomplish that goal, and then find collaborators that could add value to the project. One thing that I would advise is if you want to see who's already funded in the program, go to our website and we list all of our funded investigators. We list the title of their project, and if you click on that title, it takes you to the NIH Reporter, which is a database that has just lots and lots of information on that project, you know, from who's involved, what they're doing, and everything. And then you might get an idea of some potential collaborations to pursue.
Okay, next question. Can we propose experiments that will be done by the internal CounterACT team?
Well, I guess the best answer is we don't have an internal CounterACT team. This is an extramural program. Our intramural labs are not working with us on this. They've got lots of other stuff to do, but they're not working with on this. So I guess the answer is, you know, we don't do any research internally.
All right. The next question. I would like to learn about the range of drugs being funded and how to collaborate with Centers for Excellence.
Okay, two pieces. So the first one was the range of drugs, candidates, not the threats, from what I understand. And in that case, you know, remember, this is a long list of many different kinds of chemicals that have many different kinds of effects. So the range is pretty large. I mean, we can go from anticonvulsants to antioxidants to anti-inflammatory drugs to, you know, drugs that sequester the agent. So it's agent-based, not host-based, and on and on. So, you know, I guess the answer is, if you wanted to look if you wanted to sort of look what we're doing, that's not to say that we're confined by that, but if you want to look what we're doing and see the kinds of different drugs that are being developed, that website might be a good place to start.
Next question. What are the priority chemical threats identified by agencies?
Yeah, so as you know, we've got a couple of slides. I'm not sure what the numbers are, but if you go back through our webinar, you can see a couple of slides that address that issue. Now, if you're talking about, if you're really talking about priority in terms of the prioritized list of chemical threat agents, unfortunately, that's a classified document. But again, the main thing to do is to just give us a call, let us know what you're thinking about, let us know what the kinds of chemicals you're thinking about, and we'll be glad to help you to see if it would be feasible to put together an application.
Okay, next question. To what extent do projects need to be developed for a competitive application in the R21 and R01 formats?
So R01s, we don't support R01s. Ours are UO1s. A lot of that was covered, I think, in some of it was covered in the presentation. But I'll say this: for R21s, you know, technically they do not require a lot of preliminary data. In some cases, any preliminary data. I always tell people that well, two things: there's a pros and cons of putting in preliminary data. So the pros is you can make a better argument for scientific feasibility when you have preliminary data. The cons are, if you put that preliminary data in there, it's going to be reviewed by the reviewers, and if it's weak, you know, it could you know, hurt your score. So it's a cost. Now, I'd say this: if for R21s, I'm speaking just for R21s, if you can make a really, really strong argument for scientific feasibility from the literature, and remember, when you put that literature in there, you want to talk about the quality of that literature. that you're putting in there. This study was really good because it was blinded, and had a bunch of rigor reproducibility. This study, suggests some things, but it's not quite up to par in terms of rigor reproducibility. So, but again, if you can make a really good case with the scientific literature, it may be okay. For the UO1s, you need preliminary data. You need preliminary data as well as, if possible, data from the literature to support your scientific premise for those mechanisms.
Okay. How many times can you resubmit an application that was not funded?
Answer is once. By NIH policy, you have only the initial application and then the A1 revised application. But you can submit another grant as a new application. It just won't be, you won't have a chance to put in your introduction section where you address the summary statement.
Okay. Next question. Do you have considerations for young investigators?
Well, those of you know me that this is one of my passions. Diversity in young investigators is one of my passions. Now, the NIH has specific policies to increase young investigators within the biomedical research community, but it's only for, at least at our institute, it's only for, and I think other institutes as well, it's only for percentiled applications. Our applications are just scored, they're not percentile. But I'll say this, if you have an application that is on the bubble or close to being on the pay line but not quite close, and you want to make an argument for that, you can make several different kinds of arguments like, well, this is a really important chemical threat, and we don't have any other thing in our portfolio, and this and that. But I always include the fact that the applicant is a young investigator, diverse investigator, things like that. So we do have considerations, but not the formal ones you see with percentile applications.
Which mechanisms should small companies apply under to develop a CounterACT therapeutic and to maximize chances of funding success?
I can answer the first part easily, and that's all of them. Obviously, the SBIR program, which we do participate in. If you go into the Omnibus SBIR funding announcement, you'll see, I think it's down to just a couple of sentences now, but at one point we had a whole paragraph about CounterACT in there. So you can come through the SBIR, but you can also come through any of those programs that we mentioned earlier, the R21s, UO1s, I think, and even the centers, I think, maybe, accept applications from industry. So the second part was?
Do you have any on how to develop CounterACT therapeutic and to maximize chances of funding success?
Oh, I see. Oh, well, that's hard. I mean, it's hard to say whether or not an SBIR is more likely than a CounterACT. Both of those are scored applications, rely on the scores. It depends on funding. I mean, sometimes the SBIR has a lot of funds to support a lot of grants, and other times they don't, and the same goes for CounterACT as well. So I don't really know how to address that, but be happy to talk to that person on a one-on-one basis to try to strategize.
Next question. Are opioids toxic effects on brain related to this call, or I might have missed something?
Yeah. Well, yes, in the sense that we do have a program looking at opioid overdose. We have a notice that was, if you go to our website, you can click on the notice where we talk about the kinds of interests we have in consultation with Chris Bow and NIDA at National Institute on Drug Abuse. And the difference here is that we're interested in countermeasures that could be used after one of these toxic opioids or fentanyls is used in a mass casualty event, not really just a single overdose. That's covered by other programs at NIH. But yeah.
Are carbon monoxide and fluoroacetate agents of interest for medical countermeasure development?
Yes, they're on the list if that's what you're asking. And I don't know if I mentioned this earlier, but you are permitted to submit an application as long as it's on that list of chemicals. Whether in the final analysis, when we're deciding whether or not to fund your application, whether that guarantee, in many cases, we have to look at our portfolio and look at the distribution of different chemical agents within our portfolio. And if you have, a couple of applications with the same score, and one is on a working with an agent that is of higher interest to the Institute, that one probably would be selected. But as long as you're on that list, you're welcome to submit an application. And, we've rarely turned down applications because of the chemical selected. Very rarely.
Okay, next question. How flexible are CounterACT funding opportunities in terms of what you're able to research once you get the grant? Does this vary with different grant types?
Well, we're not flexible. So by NIH policy, you have to conduct research that has been proposed, reviewed, and approved by the NIH. We can't give you the money and then you start working on something else.
Does an applicant need to have facilities capable of handling nerve agents internally, or is there an opportunity to collaborate with NIH facilities to conduct these studies? If so, how is this budgeted?
Great question, common question. So, some of the chemicals that we study are in fact Schedule I chemicals, not Schedule I drugs, but in terms of the Organization for the Prohibition of Chemical Weapons, OPCW, are on the Schedule I list, and they cannot be used, they can only be used in approved facility, mostly DOD facilities or DOD-supported facilities. And those include things like the nerve gases, sarin and VX, and sulfur mustard, and a few others I'm drawing a blank. But those chemical weapons, they have to be done in collaboration with the DOD or a DOD-supported laboratory. And in terms of collaborating with them, again, the easiest way is to look, and there's only a handful. I'm not talking about, 20 or 30. I'm talking about 3 or 4. So you can go to our website and look at who people are collaborating with that are working with these Schedule I agents and explore the possibility of collaborations with them.
Next question. Do you also consider biological threats like viruses?
Well, I certainly personally consider them a threat. So the NIH has 3 programs, biodefense programs, basically. There is a very large biologicals program at NIAID that focus specifically on biological threats. Focus here is on chemical threats only.
Next question. I think we already touched on this about the small businesses. We can skip this one?
Yeah.
Okay. Next question. Are pre-treatment strategies viable for R21 applications? Also, are in vivo verifications required for R21 applications?
So the first question, pre-treatment strategies. So we do recognize that pre-treatments could be valuable. especially in the military, going into theater where they're potentially use chemical weapons, they've used pretreatments for years. Our focus in the civilian sector is a bit different, where we can't really predict where one of these terrorist attacks or accidents are going to happen. So our focus is less on pretreatment and more on post-treatment therapeutics. With that said, what we tell folks is that we would accept applications on pretreatments as long as it's just a small, or as long as it's a component of a project proposal that includes post-treatment. So an application that comes in that's just pretreatment is not going to be responsive to these FOAs.
Next question. Is a special study section used for the R21 applications, or are the standing study sections used for review?
All CounterACT applications, R21s, U01s, U54s, are reviewed by a special emphasis panel that has done an unbelievably remarkable job since 2006 in putting together the expertise, and you can imagine a lot of different kinds of expertise that are needed for these kinds of applications. So yes, it's not done by standing study sections. It's done by a special emphasis panel.
Okay, before we go to this next question, I just want to let you know this is the last question we have as of now. So if you do have another question, please type it in now. You can get a chance to go to the front of the line. So with that being said, let's ask this question. Can you address the success rate / funding rate for R21 and U01 applications?
Yeah, this might be a quick answer because I don't have that data in front of me. I would say it's on par with the translational research programs at our institute, which is a little bit better. And it's probably on par with the SBIR program as well. So it's a little bit better than your, usual R01 pool. But these grants are reviewed under a lot of scrutiny, a lot of different pieces that the reviewers have to consider. But yes, it's a little bit better. Our R21s, one thing about our R21 program that we're extremely proud of is the transition rate from an R21 to your U01, as far as I know, is one of the highest, if not the highest, across all of the NIH. So we're really pleased about that, and hopefully that will continue.
All right, we did get another question. Are in vivo verifications required for R21 applications?
So I think it's not required, but if you're going to make a strong argument for a U01, it would be good to have some in vivo verification. But I do not think that it's required. Shardell may not know that.
No, you're right.
Yeah.
Okay.
The reason for it.
Are strategies that relate to a wide number of threats considered more attractive?
That's another great question. Great in the sense that it sort of depends. So the general answer is yes, absolutely. That's why, we created these toxidromes, because we wanted to, and we have some products that are fairly along and that some of are already in advanced development where that would be the goal. Because as you may or may not know, one of the goals of this program, the NIH and BARDA programs, is to put drugs in the strategic national stockpile so that we stand ready and have our medical response capabilities improved. If you had, 1 million doses of one drug, and that one drug could treat, cyanide, nerve agents, and opioids, that's much more cost effective than having one for each one of those chemical threats. So yes, it's more attractive. Now, the only caveat would be when you're working, if you're working with, let's say, 4 or 5 different chemical threats in a UO1, trying to coordinate that, trying to have enough time and budget to do rigorous research for each one of those can be difficult. And then finally, when you have a drug that you're taking to the FDA, usually, and you can contact the FDA to verify this or not, usually you're going to have data for all of those threats. But the answer to that is yes, a resounding yes. It's much more attractive, especially to advanced developers who are taking these to licensure like BARDA. And I'd recommend you to look at their website as well, so you can confirm or reject what I said.
Next question. Are there any active applications close to clinical trial for synthetic opioid prophylaxis and reversal drugs at this time?
I do not know the answer to that, and I probably would have to defer to our NIDA colleagues. I would probably have to refer to our NIDA colleagues to be able to answer that, so I'd be happy to do that. But I don't know the answer to that right now.
So if the person that asked that question, if you could reach out to us, and we will make sure to direct you to the right person. All right. Now, with that being said, it looks like we've come to the end of our webinar. I'd like to thank everyone for joining us. Please do reach out and let us know if you have any suggestions on how we can make these better. You know, we're always looking for tips and suggestions. You can expect these slides in your inbox by the end of this week. In terms of the recording, like I said, we do plan eventually to have that up on our website, and we will also send it out via Twitter, and we'll even add it to our listserv. So with that being said, I'd like to, well, we have one more.
I just wanted to say thank you to Tim Lydon for running this webinar. Great job.
Thank you very much. So we, till next time, thank you very much, everyone. Take care.
Bye-bye.